Hello, my name is Ina Naumov. I'm a PhD student at Tel Aviv Sourasky Medical Center at the Department of Integrated Cancer Prevention Center, and I would like to tell you about our study, how knockdown of CD24 gene prevents colorectal cancer in CD24 APCmin double knockout mice. The prototype for the carcinogenesis of the GI tract was first presented in the early 90s by Ferron and Vogelstein and is being investigated until today. With the completion of Human Genome Project, it becomes clear that many new players in this multi-step process are yet to be defined. One such gene is CD24, which is being reported more and more in relation to various malignancies. It was found in our laboratory that CD24 molecule is of great importance in GI malignancies. In our previous studies, we were able to show using immunohistochemical staining of adenoma and CRC tissues that CD24 was highly expressed already at the early stage of the neoplastic tissue, but it was almost completely absent in the normal tissue. These findings guided us to explore its involvement and its contribution to the colorectal cancer. CD24 is a cell surface heavily glycosylated, GPI-anchored mucin-like protein. Although the protein core consists of 31 amino acids, due to post-translational modification, mainly N and O-linked glycosylations, its molecular weight varies between 25 to 75 kilodalton. The biological function of CD24 is unclear, but it was identified as an alternative ligand for P and E-selectin and adhesion receptors on activated platelets and endothelial cells, respectively. The aim of this study was to evaluate the role of CD24 in intestinal carcinogenesis using a transgenic mouse model with a double knockout of CD24 and APC alleles. APC is a key tumor suppressor gene mutated in familial colon cancer syndromes, as well as the vast majority of sporadic cases, making APCmin mice a clinically relevant model and an excellent system to unreveal the underlying genetic interactions. In this study, we adopted a genetic approach to clarify the functional involvement of CD24 gene in intestinal tumorigenesis. The APCmin and CD24 mice were mated to produce APCmin and CD24 plus pups, which were then mated again with CD24 mice to reverse the genotype of the CD24 plus to the CD24 . All the mice were sacrificed at the age of 12 weeks and assessed for intestinal polyposis according to their APCmin and CD24 genotypes. As you can see here in this slide, a deficiency of only one CD24 allele was sufficient to suppress almost completely polyp formation in the APCmin plus mice. These results were verified by histopathologic analysis. In addition to developing a large number of intestinal polyps, APCmin mice also develop a severe anemia that is generally attributed to the blood loss from the polyps. The anemia is secondary to the polyp formation. The size and the weight of the spleens indicates the severity of the anemia. As can be seen here, the APCmin mice exhibit a highly anemic spleens, whereas the double knockout mice exhibit a quite normal one, which is another indicator of their general well-being. In light of these intriguing results, we aim to understand how CD24 contributes to the suppression of the development of the intestinal polyps. Thus, we use the liquid chromatography tandem mass spectrometry-based proteomic strategy to identify new CD24 binding proteins. We report here identification of beta-catenin as a new CD24 binding protein. These results were validated and were reproducible in vitro in CD24 overexpressing cells. This is in particular an important finding as beta-catenin is regulated by the APC gene, and mutational inactivation of the APC gene is the earliest and most frequent genetic event in colorectal cancer. Nowadays, we're trying to understand the nature of this interaction. Now allow me to conclude our results. A deficiency of only one CD24 allele is sufficient to suppress almost completely polyp formation in APCmin mice that normally develop hundreds of polyps. CD24 interacts with beta-catenin, which is regulated by APC. And finally, although the mechanism underlying the pathophysiological role of CD24 in the development of colorectal cancer is still under extensive investigation, these studies suggest that CD24 plays a crucial role not only in progression of colorectal cancer but also in initiation of this process, and thus is a potential target for early detection and intervention. Now I would like to acknowledge for some people who greatly contributed to this study. First of all, my mentor, Professor Nadir Arber, and also Dr. Sarah Krause, the head of the laboratory, and the rest of the cancer prevention staff. Also to Dr. Rina Arbesfeld and Dr. Alona Zilberberg from Tel Aviv University. Professor Mina Fogel from the Department of Pathology at Kaplan Medical Center, and also Professor Peter Altvogt from the Department of Immunology at the German Cancer Research Center.